do you want to talk about the importance of developing hand signals as well?
Team hand signals?
Group hand signals.
I don't know. Dietrich and I set the bar high back when we were on the team. So, well, teams have names. You can come up with hand signals. You can come up with, chants, dances, whatever to make your team function. That should be a function at dinner. All right. Well, Will said this talk is going to be inspirational, so sorry to disappoint you. But I'm going to talk about case studies from NeuroNEXT, which, so basically I'll start, and then we're going to have a panel after this, as Will mentioned, to talk about the various networks, but just to give you a little. Is this better? Mm-hmm. Just to give you a little bit of background about NeuroNEXT to give context. So NeuroNEXT has been around since 2011, and it was really funded. You can see kind of the goals here, but in essence, it was funded to help to bring phase 2 clinical trials forward in order to better determine whether phase 3 trials were needed or not. So, kind of back at the time, pre-NeuroNEXT, there were a lot, there still is to some extent, but there were a lot of phase 3 clinical trials that were failing, to meet their goals, and kind of retrospective looking back, it was the phase 2 trials maybe hadn't been as rigorous as they needed to be. So one of the goals when NeuroNEXT was formed was to have kind of a rigorous path of passing phase 2 trials. One of the things that makes NeuroNEXT unique is that it is not disease specific. So you'll hear about StrokeNet and Siren, which have specific niches, but other than that, really NeuroNEXT covers everything else in the neurospace that falls under the NINDS umbrella. You can see here the current sites. We currently have 20 sites that are spread throughout the US. Many of you are probably at one of these sites. And so the way NeuroNEXT is structured, there are 20 sites that have funded grants to participate in the network. We are the funded data coordinating center at Iowa, and then Mass General in Boston is the funded clinical coordinating center. Excuse me. We were just recently refunded in 2023 for another 5 years. Won't talk much about this. If questions come up later, we can talk about this in the panel, but there are some new initiatives that probably the one that we're most excited about is the transition to a new funding mechanism, or the other transaction authority, which kind of is a strategy to try to decrease the time from an initial application to funding award. That technically goes into effect tomorrow. That's the first day that applications can be submitted under the OTA, so we're kind of excited about that to see how that goes. We also are adding a gene therapy consortium, and we'll be doing gene therapy studies within NeuroNEXT in the next round. There's a separate notice of announcements for that, and then you can see some of the other initiatives that are ongoing. Let me just skip that and go. But what I mostly want to talk about today is less about the network structure and more about kind of what the network has worked on in ways that might be relevant to you. Maybe not so much in the projects that you're developing here, but probably for some of you next steps as you move into more of a multi-center phase 2 space. So what I'm going to talk about here and what NeuroNEXT really focuses on is phase 2 clinical trials, which in many ways, and I've developed more of an appreciation for this as I've been involved in NeuroNEXT over the years, are some of the hardest trials to design. Because phase 1 you're looking at safety, phase 3 you're trying to show that something works. If you've done everything well, by the time you're going to phase 3 you have a pretty good sense of what trial you want to design. One of the challenges with phase 2 is you're kind of in that middle zone where you're trying to determine whether there's enough evidence to go to phase 3, so you're not really showing efficacy, you're trying to screen through. And that requires, some specific discussion and coming up with, in many cases, novel designs to try to meet those goals of what to do. So what we're talking about in phase 2 is after you've done one or more phase 1 studies, you may be initiating phase 2. They're in larger groups of patients, but not large enough to really show efficacy. And you're really looking more at risk-benefit ratio and trying to decide what to move forward. And what we'll really talk about, and what we talk about a lot in NeuroNEXT as far as proof of concept trials, is go, no-go decisions. And another thing that I've picked up kind of in working in phase 2 trials and kind of collaborating both in academia and industry is, as much as anywhere else in clinical trials, it's my opinion that phase 2 trials are where academic or NIH-funded clinical trials and industry-funded clinical trials maybe differ the most. And that when you're talking about industry development, phase 2 fall into kind of a broad program. And for instance, a company may be willing to accept some level of risk of a phase 2 moving forward based on whatever decisions have been made as part of that. The same is not necessarily true, and is often not true when you're talking about NIH-funded trials. There's often a higher bar in terms of having rigid go, no-go criteria. And if you think about that, it makes sense in the sense that it's who's absorbing the risk for doing the phase 3 trial based on the results of the phase 2 trial. If a company is absorbing the risk, they're going to pay for the phase 3 trial. It's really their decision what type of design they want to make. If NIH is going to subsequently fund the phase 3 trial, that's our taxpayer dollars. And so you would expect a slightly higher bar. So one of the things that I think that generally comes up is this go, no-go decision, in that you have to be able to think of not just how a trial will succeed, which and many of you, when you've come in and started probably discussions with your team leads, that's what you're primarily focused on. But a more important question, particularly in the phase 2 space, is how can my trial fail? And when I say fail, not fail as a trial, but fail to show that this works and is sufficient to go to phase 3. Because if your trial can't fail to tell you there's not enough evidence to go to phase 3, then it's not a good phase 2 trial design. You're basically deciding you're going forward regardless. And if you have any level of subjectivity, if you're convinced that it works, you're going to see that it works. So you want to make sure that there's some level of rigor in order to know whether it's sufficient to go forward. You also want to not just do a mini version of what the phase 3 trial would look like. I can tell you from being on study sections over the years, and I think everyone else who has been, as soon as the words, this looks like an underpowered phase 3 trial come up in study section, it's the kiss of death, right? You might as well just stop the discussion at that point, and the floodgates open and it goes on. So, and especially when you say it's an underpowered phase 3 trial where we can collect data to determine how to plan the phase 3 trial, right? Don't do that ever, right? Be clear that what is the question. And in particular for phase 2, you're thinking, what do I need to know to do the phase 3, right? What questions do I need to answer in order to do that? So, without this, and part of this gets to this issue, which is a broader thing in clinical trials, equipoise, that I think you've all probably heard about. And when we talk about that, we're really talking about more societal or clinical equipoise across the field. There is not a single investigator I have ever worked with who is an equipoise themselves. They're convinced that it works, otherwise they wouldn't be spending their career, their time, their resources in it. Hopefully all of your projects that you're working on, you're convinced that there's at least a good chance it will work. Otherwise you probably wouldn't be doing it. So almost by definition, you're not an equipoise, right? You think this works, that's why you're pursuing it. But in order to do phase 2, phase 3 clinical trials where you're asking people to be randomized, there has to be some level of equipoise across the field or some level of uncertainty as to whether this works. But if you think it works, and there is no objective criteria to determine whether the data tell you to go forward or not, you're going to see what you want to see in the data. It's the Rorschach test of clinical trials, right? Investigators will see whatever. From working with clinical investigators for years, I can tell you, clinical investigators who do research are good at 2 things. They're good at explaining why things that they thought would be statistically significant were not because something was wrong with the design. And they're really good at explaining why something that shouldn't have been significant is not important because of some type of flaw with the design, right? So be open-minded in terms of how to doing that, and the better you can put the design in place to clarify that, the better. So I'll just talk briefly about, there's a number of types of phase 2 clinical trial designs. You can see kind of a broader list here, but I'll give a few examples of things that we've done in NeuroNEXT over the years to give you some examples of how the go, no-go criteria for those are put together. And this covers kind of a broad range of diseases and types of design. So the first is kind of probably the simplest, which is just a safety tolerability design. This is when you're kind of looking at often a drug in a different population. And one of the first, so we're really probably more in the 2A space here, where you're looking at whether it's sufficiently tolerable and safe to move forward. And you may collect some efficacy data, but it's more as secondary outcomes. You're really not really looking at benefit much at all. We did this in a study in Huntington's disease to look at tolerability of a new compound. This was one of our industry, it was our only SBIR that we've done to date in NeuroNEXT. This was done with an industry partner to look at this to treat irritable subjects with symptomatic HD. Memorable to me, so just this has nothing to do with the talk, but you're going to learn little facts about me as I throw it in. But what got me through my dissertation years ago was WWE and Jerry Springer. This was a preclinical kind of Jerry Springer type model where they would put mice in with a mate for time, and then they would take them out and put another male mice in with the mate, and then they'd put the two males in and they would fight. So it's kind of like the mouse version of Jerry Springer to treat irritability, but that's how they showed this works because it calmed the mice down. And so now they were expanded into human, and you can't test this on Jerry Springer, but it was kind of the same type of experience. And so this was basically safety tolerability, where the endpoint was pretty simple. The endpoint here is just tolerability. What are the proportion of completers in each group? So if you put people on this drug over this period of time, can you keep people on that, and do you have sufficient tolerability on drug compared to placebo? Because some people are going to drop out just for random reasons. And so when this was done, so the GO criteria was basically a non-inferiority margin. This one was a wide one to do that, but it in essence met the GO criteria. So you would reject for non-inferiority, where basically you were saying that the drug was no worse than placebo for tolerability. This would justify moving this forward as a candidate to look at further, more complex studies. So that's a very simple type design when you're kind of in that early space and you don't have sufficient data on whether you could even do this. Other types of studies that we've done, and one that I think will come up more often, we want to do this in more diseases, but one of the rate limiting steps is the lack of sufficient biomarkers, but as a surrogate endpoint design. So one of the things, particularly in diseases where you have endpoints that may take a long time to see effects on, if you had a good surrogate endpoint, a good biomarker that was a good surrogate for the clinical endpoint you want to observe that you could measure in a shorter time frame, then you could potentially do your phase 2 trial based on the surrogate endpoint to see whether there's a benefit on the shorter term surrogate that would tell you to go forward to the clinical endpoint. And so here, you can often use the surrogate as your go, no go. If you're not seeing anything on the surrogate and it's a really good surrogate for the endpoint, then you're unlikely to see something on the clinical endpoint. Obviously, the strength of that design lives on the strength of how good your surrogate endpoint is or not. So you really need to have good solid evidence that it's a good surrogate. We did a study, one of the earlier studies in NeuroNEXT, this was in MS, looking at a drug called Abutalast, this was with another industry partner years ago, where obviously the ultimate goal was to look at, does this drug slow the clinical progression or treat the clinical progression of MS? But the endpoint looked at here was an imaging measure of brain atrophy. The thought was, based on the data, if you could slow the amount of brain atrophy, that would have clinical benefits downstream. And so with this, what the study showed, it actually met the GO criteria. So it showed that the drug slowed the amount of brain atrophy over a 2-year period in these individuals with MS by about 50% compared to placebo. And it wasn't powered for clinical effects, but if you look on the right with an exploratory analysis based on a clinical endpoint, it showed that it also had a trend towards slowing disease progression. So this would basically say that it would be worthwhile for the company to at least consider pursuing the larger phase 3 trial, which in order to show significance in the clinical endpoint would need to be much larger and way more costly than the phase 2 clinical trial was. Right,
So before we move on from that, so do you think that there were flaws in the design or the interpretation since it did not in fact go forward, and the narrative around Abutalast is, well, really no clinical benefit, so it didn't go forward? Full disclosure, I was an investigator in this study.
Yeah, I don't know all the discussions that have taken place since, because the company has kind of taken this since it came out. I think it probably has to do with lots of different factors. I mean, there was no clinical benefit because it wasn't powered to find clinical benefit. I think the size of the study to show clinical benefit was going to be really large and really costly. I think that may have had something to do with it. Other than that, I honestly don't know, because once it's out, it's kind of in the company's hands. And with NeuroNEXT, we don't do phase 3 trials, so the company would have to fund that, or they would have to come to NIH in order to fund that trial. So I don't know exactly what all the discussions have been subsequently to totally answer that. I know from that study it said go, but the discussions and pulling the data from other sources and making the decisions is kind of something that's multifaceted. It's a great question. Yeah. So another type of design that can be used, and this is often used when there's either decisions between multiple treatments to look at, or you're trying to choose between multiple doses or multiple ways of giving, would be a selection design. Selection designs are kind of just like horse races. There's really not a type 1 error there. You're just trying to looking for what was the best, although you can put rules in place to try to make types of inferential questions from it. One that we recently completed here was a study in Parkinson's disease looking at light therapy with Alex Vidinovich, who is an alumnus of this course, so he once sat where you sat, to look at whether once or twice daily bright white light therapy improves sleep in Parkinson's disease. This was an interesting study in that it had 2 interventions, so bright white light therapy given once daily or twice daily, so looking at that. It also had 2 controls, so once weekly bright white, which was kind of a bright white, less intensity, and also daily dim red light therapy. So and the go criteria here was fairly complicated. It was simple but complicated. So basically, in order to go here, both of the bright white light therapies had to be better in terms of the change in this endpoint, the Parkinson's disease sleep scale, by 1.7 points or more than both of the control interventions. And if they were both better, then you had an algorithm for choosing between doing twice daily or once daily, where twice daily had to be slightly better than once daily in order to go forward. What the study actually found was that it did not meet the go criteria. Interestingly, and this is one of those, often when you don't meet the no-go criteria, there's 2 paths you could go. Either clearly it shows you the drug doesn't work, or it answers more questions than or it raises more questions than it answered. In this case, it didn't, because the 2 bright white light therapy arms beat the once weekly bright white, but not the dim red light. And the challenge there was that the dim red light did not look like a control condition in the study. So that raises lots of questions. Why is that, right? Is there some effective dim red light therapy that wasn't accounted for here? Because it was pretty well accepted coming in that that would be a control. So this is one where it didn't meet the go criteria, but it's kind of raised questions. If the decision had been just weekly bright white light therapy as the control, the go criteria would have been met. So it's very confusing in terms of how to interpret this, and there's a lot of work, obviously, that is going on with the investigators to try to put that in context. Another design that I think is really useful, and this kind of MUSC has used this quite a bit as well, and I think Renee's in the back would agree this is a useful design, that is not popular because whoever named it years ago, I don't know who named this, Renee, but it's the worst name ever, because nobody wants to show their trial is futile, right? And so people hate the name. 2, people get it confused with interim futility analyses in clinical trials. And so with those 2 things, it causes it to not be as attractive as it really should be. The whole idea of a futility, and probably a better name for it, is non-superiority. So what you're really looking for in a futility design, and this is kind of a way, if you don't have other options than trying to do basically something that is structured like the phase 3 trial, to not have an underpowered phase 3 trial, to have some rigor to it. When in essence, if you think about a standard superiority test of hypothesis, we know that if you reject the null, right, you can claim that the null was not true, but if you don't reject, you don't accept the null. So in general, if you reject the null, you say there's no difference. If you don't reject, then it could be either that there was, the drug could be worse, or there could be no benefit in the middle ground. With a futility design or non-superior design, what you're really looking for is the drug worse enough that it's not worth moving forward into phase 3. So if you don't reject the futility hypothesis or the non-superiority hypothesis, that does not tell you that the drug works. That tells you that the drug may work, or there may be no benefit. You need to do a larger phase 3 clinical trial to tease that out. So it's a way to get data to help do the larger phase 3 trial while still having some degree of rigor. And there was I think Yuko Palish wrote this years ago, that they went back and retrospectively looked at a number of stroke trials that failed in phase 3 and showed that if you had done this type of design in phase 2, you would have never gone to phase 3 because this criteria would have been met. So basically, you're just kind of flipping the hypotheses in different directions. So futility designs have high negative predictive value. If you claim futility, so if you reject the null hypothesis and claim futility, it's very likely that the treatment is not effective. but it does not have it has low positive predictive value. If you don't claim futility, it doesn't tell you the drug works, but it gives you enough justification to move forward to the phase 3 clinical trial. We did a futility trial in myasthenia gravis in NeuroNEXT early on, where the endpoint here was looking at the percentage of participants that achieved a 75% reduction in their prednisone dose. So lots of percentages there, but basically the prednisone dose when they come in, the prednisone dose at the end, if it dropped by 75% or more, that was a success. If it did not, that was a failure, and then you're looking at a binary endpoint over time in a futility design where the thought was in a future phase 3 trial, the drug of interest, rituximab in this case, needed to have a 30% or greater benefit. So the null hypothesis here, which is different than your typical hypothesis in a phase 3 trial, was that rituximab improves the outcome by at least 30% compared to placebo. The alternative is that it does not. So this is very different from your typical null and alternative hypothesis, which is where kind of the non-superiority tests come from. So the null is that it is superior. The alternative is that it is not. And the go criteria here was just whether or not you found futility based on that test. You can kind of see here the power of that. So if you assume that the placebo rate is 40%, then for various percentages of the assumed rituximab rate, this is the power of the study. So if it was 70%, which corresponds to the 30% benefit that was thought to be clinically meaningful, you have 10%, which was the alpha that was used for the study. If it was 40% or less, which means the treatment is equal to or worse than placebo, you can see that there's very high power here, which means there was a high probability of declaring futility. And if it was somewhere between 40 and 70, you see various levels of power. That's the yellow zone there, which means you may or may not declare futility, the drug doesn't work. But the whole idea here is that you have high power in the red where you really don't want to move it forward. And if you don't declare futility, part of the goal of the phase 3 trial is to tease out the green and the yellow zones to try to determine whether there is a benefit or not. So it's a good screening thing with a smaller sample size in order to tell you whether it's worthwhile to take on the phase 3 trial. In the case of this rituximab study, the futility criteria was met. And you can see here kind of the curves of the two groups with the prednisone doses over time were pretty much on top of each other, which kind of reflect that. And it was determined, at least in the population for that study, that this was not worth moving forward. Right. Another type of design that comes up, this comes up in phase 3 as well, will be a run-in design where you can either use this to screen participants and randomize those who screen a certain way, or you can use it in order to basically assess compliance. This is used a lot if there's some type of diary or intervention where you need people to fill out, and the thought being if people won't fill out your diary during the screening period, they're unlikely to fill out the diary during the clinical intervention period, and you want to get complete data. An example of a study where we had this on top of a more standard design was a study in Fragile X that was completed a couple years ago. This was a study looking at language improvement in children treated with this investigational drug, where the idea behind this was that the drug would help to enhance the ability of kids to learn from this behavioral intervention to improve cognitive or communication skills in this Fragile X population. So the way this study was basically designed, there was a run-in period where everybody got placebo in order to kind of assess that there was, but the kids didn't know they got placebo. So you could assess, basically there shouldn't be any difference if people are changing, that would be a potential problem. And so then there was randomization, and then they got the language intervention on top of the randomization, where the thought being coming in that the kids who got the drug and the intervention would show greater improvements in cognitive skills over time, not cognitive, communication skills over time than the kids who got placebo. Okay. This was a language intervention that was both structured and unstructured. There was a lot of kind of interaction with the structured component. One of the complications for this study was it hit right smack dab in the middle of COVID, where structured components had to change. So there was one like blowing bubbles, which was not something you wanted to be doing during the pandemic, so that had to be changed. So one of the complications of this study, like many studies that were going on in 2020, is there had to be changes made kind of midstream to account for that. We think we did a decent job. You can never tell, right, with what the investigators came up with in terms of doing that, but also it kind of led to new ways where the intervention was done in slightly different manner than was intended at the very start. When this came through, it actually met the no-go criteria. So it turned out that we didn't see a benefit of the drug with the language intervention. In fact, the results weren't statistically significant, but all of the trends, which was consistent for the primary and all the secondary endpoints, were towards worse performance for the kids who got drug and the language intervention. So it was kind of, again, another one of those that raises, it pretty much disproved the hypothesis. Exactly why there was that consistent trend in the wrong direction was something that I think we still don't have a good handle on. Another recent study that looked at kind of a, not totally novel, but kind of an interesting design was the top spin study, which is another one that was recently completed with Gordon Smith, who's at VCU just down the street. Some of you may have flown into Richmond, so you can wave at Gordon as you fly out if you're at the Richmond airport. This was looking at topiramate, which is a common drug, whether it slowed the progression of cryptogenic sensory peripheral neuropathy, where there were two endpoints, intraepidermal nerve fiber density, which was measuring axon loss in general, and then the Norfolk Quality of Life diabetic neuropathy to measure QOL. And the way this was set up in phase 2 is that the go criteria was that the trial would move forward if there was non-inferiority on both and superiority on at least one. So it didn't matter which one you met superiority on. So if both were inferior to placebo and at least one was better than placebo, that would tell you to move it forward to phase 3. In this case, again, it met the no-go criteria. We've been on kind of a no-go trend in NeuroNEXT, which I would argue is not a bad thing. Right? It's an important value of these phase 2 clinical trials. It was no-go because both endpoints showed non-inferiority, but neither showed superiority. So remember, at least one had to meet superiority to go forward. What was an interesting component of this trial, and where it gets to another one of those where it opens as many questions as it answers, is that when you did the per protocol analysis, so this is when you look at people who had no protocol deviations, stayed in the study, you actually would have met the go criteria because, again, both met non-inferiority, but the nerve fiber density component actually showed superiority over placebo. So one of the things when we've dug into the data there, what it looks like, and again, this is something that is a little difficult to wrap your head around in terms of exactly what it means and how you would use this long term, is that if you excluded people who couldn't tolerate the drug worked, right? But part of the challenge of what tolerate the drug meant here is not really bad things. And so it's a question of, is it something education could overcome? It was something that the DSMB was very interested in early on. It was coded as cognitive disorder, which sounds horrible, right, in terms of the MedDRA AE term. But it was mostly lightheaded, dizziness, just feeling off, and mostly things that resolved within a few weeks. So in hindsight, right, if you knew that, one of the questions that came out of this is if you knew this and you could counsel patients, you may have this may go away in a month, if you could keep people on it, would the drug work? That trial hasn't been done. That's a whole question of whether that would be helpful or whether that would not, but that's another thing where the question comes. We do ITT, we do per protocol. If you guys don't know what those terms are, ask. You'll run into that. Often we do that to confirm the results because 90-95% of the time the per protocol and the ITT give the same results. When they don't, that's when you really want to dig in and ask why. A couple of recent ongoing studies are kind of whole different animals in NeuroNEXT. We have one in GNE myopathy, the MAGIN study, which is looking at efficacy of Menac. It's this powder that you give for kids in GNE myopathy, which looks at the ability of that powder to slow progression of muscle strength decline to placebo. The endpoint here is this change in several different endpoints, which is based on this model of disease progression that we're working on with Melanie Quintana, who's with Berry Consultants, who is like in the very back, our Berry group here, who developed the disease progression model, which basically is a way to do trials like this with smaller sample sizes than you would need for the standard clinical endpoints, which makes this a doable study. The other study that we have going on is the Extinguish study, which is looking at NMDAR encephalitis, or those of you who've seen Brain on Fire or read the book or on Netflix, it's that disease. This is looking at the ability of inebilizumab to treat, where we're using like a modified version of the modified Rankin scale. which is basically the modified Rankin scale in stroke that's been modified to treat encephalitis. So there's a lot of modifieds being thrown around there. One of the interesting things about both of these studies, it's both interesting and challenging, is remember I said NeuroNEXT does phase 2 clinical trials. These are rare diseases. And so as I like to say, these are phase 2 trials until they ain't. So when you think about this, we're kind of like the Scooby-Doo gang, where we're looking at these studies and they're a phase 2 study, right? And if they don't meet the endpoint, so they don't show the drug works, they will forever be a phase 2 study. But the minute that they're positive, both of these are going to become a registration trial, right? I mean, both companies have already said as such. So what is really challenging about that is you're running a clinical trial that is funded and resourced as a phase 2 clinical trial, but that needs to have the rigor and structure to potentially be a phase 3 clinical trial, which is kind of an awkward line to straddle, right? If you go too far for that, you're going to pour a lot of resources into something that, if not positive, will never need that structure. If you ignore it and the trial is positive, you're going to maybe run into complications with getting it approved by FDA when you go because of flaws in what you do. So these raise interesting challenges and discussions, and it's important, I think, with both of these to have partnerships with the industry partners who recognize some of these complications and move forward. With, for instance, next week with Stacy Clardy's study, the encephalitis study, we're meeting with the industry partner in Utah to try to sort through, some of these issues and make sure that all groups are on board in terms of how to do this. So it's an interesting challenge in that, but it's something in the rare disease space. I mean, basically, if these trials work, it would almost be impossible to do a larger version of them. And there aren't options for that. So the evidence should be strong enough, but that's something that you have to think about, right? And so when you say these are phase 2 trials, they're kind of phase 2 trials, right? That may pull the face off at the end like the Scooby-Doo villains always do, and prove to actually be a phase 3 trial. So another just complication for any of you working in rare disease. Yeah, do you have a question?
What is a registration trial? What is the actual?
A registration trial would be typically like in a phase 3 trial if you're developing a new drug, when you would do it with the intention to submit a new drug application to FDA to try to seek approval. So in other words, usually you wouldn't do that after a phase 2 trial. Your phase 2 trial would inform the phase 3, which would be more of your registration trial. What this is saying is phase 2 trial, if it's successful, there will never be a phase 3 trial. You would probably use that data to submit an NDA to try to seek approval from FDA. Does that answer? Yeah.
So when you design something like this, how do you not fall into that trap of being an underpowered phase 3?
That's a good question. I don't think one of the ways to get around that for these is nobody knows what the endpoints are, so nobody knows if it's a powered or underpowered phase 3 trial because nobody really knows how to design them coming in. So that's one way I would argue it. Other than that, I think even coming in from the start, you kind of have to know that these are a different animal, right? Like, in other words, this disease progression model is probably, if you believe in it, right, which we do, and I think FDA does based on discussions, but there's a little hesitancy in it. If you believe in it, then it is kind of a definitive endpoint, and these are kind of like phase 3 trials, but there's not enough evidence to justify this being a phase 3 trial up front. So part of that is, with these types of studies, it's pretty common to have a meeting with FDA even before the phase 2 trial to get feedback on what might need to happen. I know I'm really giving you an obtuse answer to your question, but there's really not a good answer to it.
Hey, Chris?
Yeah.
Another answer?
Yeah, Kirk, my answer.
Given this is so rare a disease, I would say odds of anybody actually running a bigger phase 3 than this are so small that the FDA is probably just stuck in the situation of they have to accept this, or they're not going to get anything bigger or bigger.
But they won't tell you that up front.
No, they absolutely, but
Yeah.
you kind of...
Yeah.
But you have to talk to them.
Right.
In the very, before you even start the study, right? You need to say,
Yeah.
They have to be involved.
Well, what you want to do is talk to, because they won't tell you, right, to
Right.
Kurt's point, but they will tell you what not to do. If there's something they adamantly disagree with, they will let you know that. So you can kind of read the tea leaves. in the room.
Or they will tell you, you know, if you're serious about this, you should use this outcome measure.
Yeah.
That's pretty telling. That would be something that they would find important, right?
Yeah. I mean, there's no, you're never going to know for sure. You just want to make the case, and you want to be as careful as possible in doing it, that you do it in a way that will live up to the rigor needed to justify it. But I saw another hand up somewhere over there.
And is that true in an agent like inebilizumab, which is already approved for another indication, or you're saying that could be the case in a novel agent as well?
It could be the case for either. So in 111, it's using an agent that's already approved. In 109, it's not. So, you know, there's two different examples of where each could be the situation.
And this agent is not like a nutraceutical. This is an actual pharmaceutical. This is
Yes. Yeah.
And just add that the distinction here is whether you're looking for a new indication, so it's not a new drug.
She said they're both looking for new indications. Is that what you
As
said?
opposed to a new drug,
Yeah.
it's about new indications that then lead to the substantial evidence of typically 2 trials or more data.
but this one is a drug.
This one is a drug, yeah. Any other questions? All right, well, the last thing I will say before we move into the panel, because I think this is really relevant for all the networks, one of the most common misconceptions with the networks is that you have to be a member of the network to submit a proposal for a trial to the network. And that is not true. It's not true for StrokeNet, right? It's not true for Siren. It's not true for NeuroNEXT. That anyone, anywhere can submit a proposal. So basically all the networks, you know, they have their different criteria for what they cover, which you'll hear about in a minute, but they're all looking for the best science. So for all of you, you know, which network might fit best is probably something that, you know, you maybe get a better sense of after the discussion next. But if you have a good proposal, whether you're at one of the sites I showed here, whether you're at a StrokeNet site or a Siren site, good ideas, good scientific ideas are welcomed by all the networks. So with that, I'll stop, and I think we'll move into the panel.
Any questions? Other questions?
Are there other questions? Yeah.
I think that maybe you already answered this, but so the NeuroNEXT funding pathway is not meant to be a bridge to other definitive funding for the trial. It's meant to actually fund the trial.
That's correct. Yeah. So it would fund. So the idea, like, if it funds the phase 2 trial, there's no guarantee of funding. Like if it's successful for phase 3.
Right. With that.
But if it's accepted, that would be a discussion with an INDS. There's a separate phase 3 mechanism for an INDS that you would apply for, you know, based on the data, the preliminary data from your phase 2 trial. Anything else? All right. If Jerry Springer reruns come on, watch it sometime.
Is this gonna be like Jerry Springer?
Especially Jerry Springer pre
Throw a
when they
chair.
had to stop fighting because of all the controversy. You know, in the '90s when they fought all the time, that was the glory days of Jerry Springer.
Yeah.
And Will's from Cincinnati, where Jerry Springer was the mayor.
Yeah, I was there when it was mayor.
So we're going to segue to the panel discussion on the three networks. And we just heard a